Yeah, I mean they're pretty variable stories, right? I mean they're out there. So we haven't started to do our studies yet. But I mean the reports are fairly. There's a lot of commonalities, and something that pops up a lot in the literature on high doses is having some sort of contact with what seems to be an entity that is potentially freestanding and autonomous in its own right in a different dimension or something like that. And that's pretty common. It pops up a lot. Uh, there's some debate whether that's potentially a cultural thing. And that's a very interesting question in and of itself. I remember I did a talk not too long ago in Arizona, I think it was in Phoenix, and somebody asked a really good question that I think I gave some smart ass answer because I was like, man, I don't have an answer to that. But it was regarding like with people that speak different languages, has it ever been kind of like investigated what the sort of link, like what language was being communicated by, because communicate like two-way communication with entities is very common in the DMT experience. So something like that I think would be an interesting question that would kind of hint at like the cultural significance of it. um but I mean, yeah, bizarre stories, but I think a pretty common thread seems to be entity phenomena. And yeah, so I mean, I'm just kind of dipping my feet in that world now and starting to understand more of the literature on the experiential components, because I was living in the molecular biology world for so long with the endogenous stuff, and now that we're ramping up to be administering DMT in humans, yeah, we wanna understand everything related to safety and proper dosing, and then also the experiential component is going to be huge in the investigation. So we're very interested in the types of experiences that people have, particularly in the domain of like entity contact and things of this nature.
I
And then
have
just really basic science questions like how does, I mean, we have the potential to really change the visual system, right, with DMT, and at a basic level understanding like what happens in the brain to juxtapose that with just like normal visual processing is something that we're interested in doing. And then of course, we a lot of the studies we do at UCSD are geared toward alleviating chronic pain. We
mm,
have a very large pain research community here, so we're particularly interested in things like I was saying earlier, like DMT for stroke and other pain conditions that don't have a lot of don't have a lot of options. So yeah, so it's a pretty big study, and out of the gate it'll be in healthy individuals, but yeah, we hope that it can help some people for sure. That's the main goal, and then to do the cute neuroscience in tandem.
this is a 2019 paper from the University of Michigan, which found one that DMT's level in humans is comparable to serotonin and dopamine. The two enzymes that are responsible for DMT synthesis occur in the same nerve cell. And three, cardiac arrest was induced in rodents in the study, which led to a 600% increase in DMT in the visual cortex of the brain.
Sure. So let me try to go one through three. So the first one, so we didn't actually measure DMT in humans in the
Mm,
study. We measured enzymatic expression. Well, the mRNA, so like it goes from from the mRNA to the protein. Um, and so we did see pretty high expression for the first time in human tissues, human brain tissues, and the frontal cortex, I think, in human brain we found, and in the pineal gland also, of the enzyme that takes tryptamine to dimethyltryptamine. And okay, so that was one. And then two, I think you said the colocalization. So there's two enzymes. And one of them is an enzyme that takes tryptophan into tryptamine. It's like a decarboxylase enzyme, and it's AADC for short. And that enzyme has been known to be present in brain tissue for a long time. And then the other one, INMT, is the one that we saw in the human brain tissue. So in the rats, in the rat tissue, this is all post-mortem. So by the way, so there are tissue samples that have been preserved after death, and then we're able to do some molecular biology techniques to amplify the mRNA that's in the tissues. So what's cool about that approach that we took is it's called in situ hybridization. You're able to go by like a cell by cell level, so you could look within individual tissues and individual cells within those tissues. And so that's what we did. So in the humans, we were a little more limited on the samples that we had. And we also, unfortunately, didn't have the two probes for the human tissues. So we only looked for the INMT. And then in the rodent, this is point two that you were alluding to. In the rodent study, in the rodent tissues, we found in the same cells in the brain, presumably neurons. We didn't have neuronal markers, but they looked pretty neuronal in their architecture. And we saw in the same cells, we found INMT and AADC, which would increase the likelihood that cell would be able to make DMT. So that's point two. And then three is the increase at cardiac arrest. Yeah. So yeah, I mean, unfortunately, like when you're doing animal research, at the end, you euthanize the animals. And so we were given permission to keep monitoring and measuring throughout the death process. And we did see, following cardiac arrest for upwards of 60 minutes later, that the DMT levels increased upwards of like 11-fold, actually. So even more than what we reported, because we had a bunch of different strains of rats, and we wanted to keep it as apples to apples as possible, and look at just within one strain of rat, because we did see some differences in different strains in terms of like neurochemistry, which is a completely another thing, but interesting. But anyhow, yeah, so I mean, we don't know though, like what that means, because it's just a fold increase. So you have to like keep in mind like baseline. Just because something goes up 11-fold doesn't necessarily mean that so much if it started out low. So the nanomolar concentrations that we reported, that was kind of like novel for the study, because again, like no one thought that it was in like the same nanomolar range as serotonin. And in the dying brain, serotonin increases like big time at death, at least in rodent models, way more than DMT. But it's interesting. So like there's a couple of papers from the same group that were actually like before we put out the 2019 study. So the Borgigan lab, Gimo Borgigan at UMich, where they looked at neurotransmitter changes and electrical changes in the dying brain. And we did too with the DMT study. So, but one of the take homes, I think, is that it wasn't just like everything increased, right? So like we monitored a whole slew of different neurotransmitters and neuromodulators and neurocompounds, and it wasn't just like everything, just like the cell like dumped everything out, right? So it seems like it's a crude speculation based on not a lot of data, but it seems like that it's like a regulated process dying, right? Yeah. So Rick, I think he lays it out pretty clear that he's speculating, right?
Yeah.
He says if one were to wildly speculate, and then he proceeds to be the one to wildly speculate. It's very Hunter Thompson of him. But I mean, I don't know. I mean, I think it's important to think big like that and to have some theories that are exciting, and they could potentially be dead wrong. But it inspires people to have imagination and creativity. It's like it's so lacking in science, kind of, because everybody thinks it has to be rigid and mechanical and materialistic, which I mean are sort of important qualities for science also. kind of keeps things grounded a little bit. But so we're not there yet, clearly, and we're not there yet in terms of being able to answer those questions. I think that it's an interesting hypothesis, and Rick and I have talked about this to a pretty large degree, that there could be, much like we talked about the endogenous opioid system, the endogenous cannabinoid system, there could be a system that isn't necessarily the serotonin system that is engaged by psychedelics. And a prime candidate for that system would be an endogenous DMT system, right? Because it's a molecule that has psychedelic properties that exist naturally. So if we understood more about how that compound exists in our bodies and what its physiological role is in our bodies, that's kind of step one, before asking the bigger questions. I don't know. I mean, yeah, imagine, speculate, and keep it grounded. Step one is to do the really foundational stuff. But if that research doesn't take place, and then you can't push it forward anymore. So it would be very unfortunate if more groups weren't looking into why the body makes molecules that have psychedelic properties, at least in large amounts of exogenous administration. It's a very fascinating question.